I'm Jim Comolli, and I'm group leader of the Biomedical Microsystems Development area here at Draper Laboratory, and an investigator working on developing nanoparticles to trap respiratory viruses. The initial aim of the project is to counteract influenza virus, which, as you know, can cause severe respiratory symptoms, fever, aches and pains, and nausea that can last up to 1 to 2 weeks in normal individuals, and it can be even more of a problem in elderly patients, where it might require hospitalization or even cause death. Another concern with influenza is the possibility of a strain which can spread worldwide and cause a pandemic. This was most recently seen in 2009 with the H1N1 swine flu, which infected tens of millions of people throughout the world. It didn't really cause an increase in hospitalizations or deaths, luckily. However, previous pandemics, such as the 1918 Spanish flu, did, and that actually killed tens of millions of people worldwide. Influenza's normal host is avian species, both domesticated and wild, and there's many strains that are circulating in bird species at all time. The problem is that when one of these species develops the ability to transmit from human to human. And recently circulating strains, H5N1, H7N9 that you may have heard of, which are of concern, and scientists have been monitoring these strains very closely. Another concern is that people can use the knowledge gained or use these strains as a bioweapon that can be intentionally released to spread throughout the population. In order to explain how our nanoparticles trap virus, I'm going to have to give a little bit of background on the viral life cycle. So viruses themselves are only quasi-living, and they have an outer coat or shell that's composed of proteins and sometimes lipids. Inside of that is the genetic material for the virus that encodes all its structural components. So when a virus interacts with a cell in the body, it is taken up by that particular cell, and then its genetic material is released. And then it actually uses the machinery of the cell itself and then make more of the components that make its outer coat. And then those are put together, and many virions are produced, and then those exit the cell and are able to infect other cells. Another important aspect of it is the copying itself is slightly inaccurate, so that the types of virus coming out are slightly different than the one that enters the cell, and therefore you get a population of viruses that come out, and this enables the virus to adapt and evolve. Influenza virus enters your body, and then it can bind onto the cells of your airway. It enters those cells, it can infect those cells. Many influenza virions are produced. They spread to surrounding areas of your lung and other parts of your body, and then they also stimulate your immune system, and this is what causes all the symptoms and detrimental effects of the infection. Current countermeasures for influenza include vaccines and drugs. Vaccines are designed so that they prime your immune system, so when it sees an influenza virus, it can block that virus and prevent it from infecting your cells. That's a very specific interaction. So in order to make a vaccine, people try to predict what influenza strains are going to be circulating for that particular season. The issue arises when, because of this viral evolution and because the possibility that pandemic strains emerge, the vaccines which are available often don't match well with the strains that are problematic or circulating, and this happened actually in 2009. Antiviral drugs are designed to inhibit a part of the process or life cycle of the influenza virus, because it has this ability to change and adapt, change its processes slightly so that it no longer is susceptible to that drug, and this is called resistance development. We've taken into account the limitations of these current countermeasures in developing our viral nanotraps. So what our traps do are prevent the virus from its initial interaction with the host cells. In the case of influenza, we know that its initial interaction is with cells of the upper airway, and strains which are able to infect humans, or human-adapted strains, bind onto particular structures on the surface or molecules on the surface of these cells that are composed of sugars. They're called carbohydrates, and basically there are chains of sugar subunits. So our collaborators at MIT have identified a particular structure of this carbohydrate, which is common amongst all human adapted viruses. So all human-adapted influenza strains are able to bind onto it. So what we do is we manufacture it, attach it to a lipid nanoparticle or a liposome, produce these in an aerosolized formulation that would be delivered to your airway. It would be able to coat your airway and then act as a trap or coating that would not allow viruses to penetrate the mucus and get into your airway cells. Instead, they would bind onto these traps and be cleared and swallowed with your mucus. What we've done so far is we've done the manufacturing, we've synthesized the nanoparticles, we've demonstrated that they work as we say they would, meaning they bind onto the influenza viruses and are capable of doing that, and then that they can prevent infection of cells in tissue culture and in an animal model. In this case, we use a strain of influenza called H1N1. And as you can see, when we mix the H1N1 with just saline or buffer, all the mice die within 8 days, and this is shown in the black line. When you mix the virus with liposomes that are lacking or lipid nanoparticles that are lacking any carbohydrate on the surface, you get about the same trajectory, and this is shown in the red line, where they die in about 10 days. However, when we use liposomes that have the carbohydrates on the surface, you can see that about 40% of the mice survive, and that those which do die actually live for a longer period of time, so it increases survival. The next steps are to improve our ability to manufacture or make these, further demonstrate safety and efficacy of an actual formulation that's aerosolized. Another aspect of our research has been demonstrating that this is a broad spectrum platform that can infect many different types of viruses. We know that the interaction of not only influenza virus, but other virus classes with host cells relies on glycans or carbohydrates on the surface of those cells. What we've done is take another type of glycan or carbohydrate, attach that to lipid particles, and shown that it can inhibit another class of viruses that contains respiratory syncytial virus, which is one which can be very serious for children under 2 or the elderly. Nanoparticles are really composed of just lipids, which are normally found in all cells in your body, and we use those which are natural to your body, and carbohydrates, which the components of are very well tolerated. So we believe that between those that we can produce something which is very safe and efficacious. Initial interaction of virus with the nanotraps, if the virus changes and evolves away from making that interaction, then it won't be able to infect its normal host cells, so therefore it will be less able to cause disease. With those things in mind, we believe that we have developed a new approach to counteract all different types of viruses, many different types of classes of viruses, and to preempt their spread if they're retentionally released or if there's a pandemic strain which emerges. And we can develop this very quickly and have it implemented fairly rapidly throughout the population.